Hello everyone. Today's topic is food microbiology. Uh, Everyone may know what is a food microbiology, but if there is anyone who don't know what is a food microbiology, let me explain this to you. Everyone knows what is food, but talking about microbiology, it is basically study, okay, study of living organisms, okay. What is microbiology? It is basically study of living organisms. What do I mean by living organisms over here is the microorganisms. Okay, so when we study these microorganisms, it is called microbiology, and whenever these microbes are related to food, it is called food microbiology. Simple as that. Now, uh, what actually are those microorganisms that is concerned to us in terms of food microbiology, right? I mean, what are those organisms that we have to study in terms of food microbiology? Now let's see them. Okay, we will be studying about fungus. Okay, we will be studying about bacteria. Okay, and we will be studying about virus. Not only that, last but not the least, we will be studying about protozoa. Okay, so these actually are the organisms, say microorganisms, that we will be studying about. Talking about some of the general concepts about this one, fungus, we will be actually studying this one, yeast and molds. Okay, so these two fungus we will be actually focusing on particularly, and protozoa we will be focusing only on parasitic protozoa. Okay, okay, simple as that. Okay, we'll be studying about fungus, bacteria, virus, and protozoa. These are the microorganisms that are concerns to us. Now, going to the next page, uh we will be starting with bacteria. Okay, we will be studying about bacteria. Everyone knows that bacteria is basically single cells. Okay, so what is bacteria? It is basically single cells. Okay, and see, though bacteria have spores, okay, bacteria have spores, but these spores are actually not used for reproduction. But they have spores, but it is not used for reproduction. Okay, simple as that. Now, not only that, so how do these bacteria actually reproduce? They actually reproduce by what? Anybody can say? Fission. Okay, so bacteria actually reproduce by fission. What is a fission? It is splitting into two. Okay, whenever any nucleus splits into two, it is called fission. Simple as that. Fusion means mixing. Fission means splitting into two. Okay, simple as that. Okay, even the yeast also reproduce in the same way. Okay, like it means same way means fission. Okay, by the process of fission, yeast also do reproduce. Now we will learn about some of the bacteria groups. Okay, uh bacteria groups. Okay, we will be learning about some of the bacteria groups. We will just note it down first. Okay, yeah, so we have to study about I mean we have lactics group of bacteria. What are they? We will actually discuss. Okay, we have to acetics. We also have beatrix. Okay, we also have propionics. Yeah, propionics. Yeah, so last but not the least, we have proteolytic bacteria. Okay, proteolytic bacteria. Okay, now what are these lactics, acetics, bitrix, and propionics and proteolytic? We will discuss about it, don't worry. Now this lactics, it comes like can you like see the similarities between lactic, right? Lactics, lactic, right? So what is this lactics? It is basically lactic acid forming bacteria. Simple as that, right? What is lactics? It is actually a bacteria. Okay, lactics is a bacteria, that is lactic acid forming bacteria. What do I mean by that? Means this bacteria actually gives a product of lactic acid. Okay, whenever these bacteria act upon any products or any items, the product that it gives, okay, that is called the product is actually lactic acid. Simple as that. What about acetics? It is acetic acid, okay, forming bacteria. Okay, what are butyrics? It is butyric acid forming bacteria, and same applies to everywhere. Okay, propionics means propionic acid, propionic acid forming bacteria. Simple as that. And proteolytic bacteria means we yeah. So proteolytic bacteria means they actually produce proteolytes. Okay, simple as that. Nothing much to know about that, right? We will discuss about it in the next section. Okay, so these are the things that I mean, or say these are the bacteria groups that we have to know. Now we will discuss each of them in detail. Okay, now let's start with uh yeah, so we can start with lactics. Okay, okay, so this is lactics. We will start with lactics. Now talking about this lactics, as I have already told you that their product is actually lactic acid, right? Now, so what is the product that this lactics bacteria will act upon? They will act upon sugar. Yes, you're right. So these lactics, they will actually act upon sugar, and they will convert into lactic acid. Okay. Now these lactics will act upon sugar, and the results will actually be lactic acid. Simple as that. Right. I mean, how do they, what is the that process of acting upon called as? That is called as what? Fermentation. Okay. When sugar undergoes fermentation, the product is lactic acid in the presence of lactic acid bacteria. Simple as that. Okay. Okay, am I getting it? This one? Okay. So whenever this lactics acts upon sugar and undergo fermentation, the product is lactic acid. Okay, now one very important thing that you may know, okay, that is related to fermentation, and that is something like this. Okay, yeah, science of fermentation. What is this called as? Can anybody say what is science of fermentation called as? It is called as zymology. Zymology. Okay, so science of fermentation is actually called as zymology. uh, since we are studying about fermentation, I thought that this will be actually important for you. Okay, uh, okay, okay, we are done, we are done. Now so what actually means fermentation? If somebody is still there who don't know what is the meaning of this fermentation, let me tell you that fermentation is basically breaking down into simpler ones. Okay, fermentation is basically making other things simple. Okay, I mean breaking down into smaller parts or whatever it is. Now these lactics, okay, these lactics, where it is used, this is basically used in preparation of sauerkraut. Okay, sauerkraut and cheese preparation. Okay, this lactic bacteria, or say lactic acid forming bacteria, is used in the preparation of sauerkraut and cheese preparation. Simple as that. Now let's start with the another topics, that is ascetics. Okay, ascetics. Now, as I have already told you, that ascetics are those bacteria that give a product of acetic acid. Okay, means they are acetic acid forming bacteria. Particularly about acetics, we will actually focus on two genera. Okay, two genera of bacteria. Now what are those two genera? One is uh, we will study about or say concern about acetobacter. Okay, and the other is gluconobacter. Okay, now these are the two genera of bacteria that is concerned to us from acetics group. Simple as that. Now what do these acetics actually do is, they oxidize. Okay, they oxidize alcohol to what? Acetic acid. Okay, now these two bacteria, they oxidize alcohol to acetic acid. Simple as that. Last but not the least, we also have this, what do we say? Beatrix. Butyrics, right? Beatrix. Now, these beatrix, what are they and what are their characteristics? They actually are spore forming. Okay, means these bacteria, they form spores. They are spore forming. Not only that, they actually are anaerobes. Means they live in the absence of oxygen. They don't need oxygen to survive. Okay, so these are the beatrix characteristics. And the most important genus of beatrix is clostridium. Okay, okay, okay. Beatrix is actually spore forming bacteria, and they actually are anaerobes, and the most important genera of beatrix is clostridium. Okay, okay, simple as that. Last but not the least, we also have what do we have right now? We have one more, that is proteolytic. Okay, proteolytic bacteria. Okay, we will be learning about this bacteria right now. Okay, so this proteolytic bacteria can be either aerobic as well as it can be anaerobic. Okay, means some proteolytic bacteria can survive in the absence of oxygen and some can survive in the presence of oxygen. Simple as that. Now the most important say bacteria of proteolytic group is Pseudomonas fluorescens. Okay, so this actually is the most important bacteria from a proteolytic group. Not only that, these Pseudomonas fluorescens are also non-spore forming. Okay, okay, these Pseudomonas fluorescens are also non-spore forming. This is very simple. There is nothing much to focus on, right? Okay, one more concepts that I want you to know is this one. The term putrefactive. Putrefactive. What is the meaning of this one? If anybody knows, please comment in the comment section below. Okay, so what is a putrefactive? I will tell you right now. Now, what actually happened is that whenever any protein undergo decomposition, okay, whenever any protein degrades or say decomposed under anaerobically, means in the absence of oxygen, it is called putrefactive. Means if I write it in simple words, it is a protein decomposition or say a decompose anaerobically. Anaerobically, okay, and this process is called as putrefactive. Simple as that. Okay, now when any protein undergo when any protein undergo uh, decomposition in the absence of oxygen, it is called putrefactive. I hope you will not forget this one. Okay, now last but not the least, we still have this one, lipolytic bacteria. Okay, this is very important. Please do understand it and understand it properly. Okay, so we have lipolytic bacteria. Now what is this bacteria actually special about is something like this. Okay, as you can see by its name, okay, it is a lipolytic, means what? Lipase. Can you just like remember like lipolytic lipase, right? So this bacteria actually produce what? Lipase. Okay, this bacteria produce lipase. So what is this lipase actually doing? This lipase will actually increase the reaction. What? It will actually increase the reaction. Which reaction I'm talking about here? Hydrolysis. This lipase will act upon fats. Okay, this lipase will act upon fats and convert it into fatty acid and glycerol. Okay, what? This lipolytic bacteria will produce a lipase. Okay, this lipase will act upon fats, and these fats will hydrolyze. Okay, will undergo hydrolysis, and these fats will convert that into fatty acid and glycerol. Okay, am I right? Now this, what is a function of this lipase over here is it increases the reaction, or you can say it catalyzes the reactions of hydrolysis. Okay, that's very simple. Now what is hydrolysis? You know, it is basically uh, uh, reaction in the presence of water. Okay, everyone knows that one. Now we still have one more, and that is okay, what do we say? Saccharolytic bacteria. Saccharolytic bacteria. Okay, we'll be learning about this saccharolytic bacteria right now. Now this saccharolytic bacteria particularly acts on, you know, those disaccharides, or it also acts on polysaccharides. Okay, okay, now this saccharolytic bacteria actually acts upon uh, disaccharides or polysaccharides, and it converted them into, or say breaks down into what? It will convert disaccharides and polysaccharides into simple sugar. Okay, so this is actually the functions of saccharolytic bacteria. Last but not the least, this is in fact the most important, I mean not important but interesting part, I must say. Okay, this is actually the pigmented bacteria. Okay, how should I write? Yeah, pigmented bacteria. Okay, so what is this pigmented bacteria? As you know, it is like bacteria who have pigments, and because of which they have different colors. Okay, everyone knows that one, right? Yeah. So what are the examples of those bacteria which are pigmented? We have Flabobacterium. Okay, we also have Serratia. Okay, and not only that, last but not the least, we have Hello Bacterium. Okay, so these are the bacteria which are actually pigmented, and obviously they possess different colors, right? So what are those colors? Come on, let's see this one. Okay, Flavorbacterium can be of a color. Yeah, Flavorbacterium can be of color like yellow to orange. Okay, now this Flavorbacterium can be actually yellow to orange. And talking about Serratia, it is actually red in color. Okay, and talking about Halobacterium, it is pink in color. Okay, simple as that. So this is like very interesting, in fact, you know, part of this one, right, from this food microbiology part. Okay, now if you see anytime, like if your milk is being rotten, or if you're getting like a foul smell from the milk, it's basically because of two bacteria involved there. Okay, if you, yeah, so that, what are those two bacteria? They are streptococcus and lactobacillus. Lactobacillus. Okay, so these two bacteria actually are involved in the uh, producing of, or say production of a foul smell in your milk, and they will actually give you symptoms of, you know, this slimy or ropey appearance. Okay, whenever you like feel that your milk is rotten and there is no smell, or say there is a foul smell from the milk, there is because of two bacteria, okay, and that two bacteria are streptococcus and lactobacillus, okay. And the symptoms of, like, you can say, like, there's a infections of these bacteria. How can we say? Because of the appearance of the slimy and ropey milk, okay. Simple as that. Last but not the least, very important, bacteria, okay. We have this one. What is that? We can change color, I think. Okay, okay. uh okay, we will try with this one. Okay, so we have here a coliform bacteria. Now, what's special about this bacteria? Okay, we will see. Okay, so this coliform bacteria, they actually are what? They are aerobic, means they need oxygen for their survival. These are coliform. Not only that, they are gram negative. Okay, they are gram negative. Last but not the least, they are also non-spore forming. Non-spore forming. Okay, so coliform are the bacteria, okay, that are aerobic, gram negative, and non-spore forming. I have tried my best to make the concepts as simple as possible. I have tried to make it the complicated one so that people can understand. I mean, in a short time. Okay, I have like compiled all the details that are being mentioned in the book, and I have tried my best to compile it. Okay, I hope you will like it. Now we will actually be studying about moles, okay. This is a very interesting part, I must say. Okay, we will be discussing about what? We will be discussing about moles. Okay, so what is moles? I have already told you that moles are a type of fungus. Okay, yeast also is a fungus. Now, talking about these moles, these moles, they actually like constitute about 25% of Earth's biomass. Okay, these moles actually constitute about 25% of Earth's biomass. That is not less. Okay, moles do have a good populations in the Earth, right? And if any food is being contaminated or by moles, you can see over there some sort of like cottony appearance. Okay, now this is a symptoms, okay, whenever any food is being infected, or say yeah, by molds. Okay, now if there is any like sort of like cottony appearance in the food, you can say there is a mold in it. Okay, right, the food is being destroyed by molds. Not only that, we also have some like usefulness, or say utilities of these molds, and that is making, or say preparing oriental foods. Okay, making oriental foods. Now, what is this Oriental foods? Basically, it is like Asian style cooking. Okay, I must say Oriental foods are like Asian style cooking, and they are like soy sauce, miso. These are the foods that we can actually prepare by the use of molds. Okay, molds are not only uh, bad for us, but it can also be used in some positive ways, right? Yeah, there is always a positive way out. If we can see even in negative part also, right? There is always a positive one, right? Okay, so these moles, what actually these moles actually do is that they actually produce what? One toxin. What is the toxin name? That is actually mycotoxin. Okay, now this is a toxin that is produced by moles. What is that? That is mycotoxin. And where is this mycotoxin found? They are found in like animal feed. Okay, they can be found in animal feed and animal products. Okay, they can be found in animal feed and animal products. Do it means that moles are from animals? No, moles can actually be found in like wheat bran, okay, and not only that, it can also be found in maize grain, okay, maize grain. It's very simple, right? Mycotoxins are being produced by moles. Moles is a type of fungus, okay, and these mycotoxins can be obtained from or say found in animal feed, animal products, and wheat bran, maize grain, and these are the simple things, right? Okay, one more very important thing I want you to learn is about this is very interesting. Okay, telephytes. I mean, I was like, I like these telephytes actually. Of since my graduation, I have always like these words actually. Right. So, what is this telephytes? Telephytes. Yeah, everyone may know or some may not know. Okay, what that doesn't matter, right? You'll be learning over here, right? So what is telephytes? Telephytes, they actually are tellers. Okay, as you can see by the name, what is okay, they have actually tellers. What is tellers? It is actually a vegetative body. Okay, what is this? It is actually a vegetative body. Tellers. Okay, and these telephytes, they actually don't have what they don't have true roots. Okay, they don't have true roots, or they don't have leaves also, or say sorry, they don't have this one. Yeah, so they don't have leaves also, and neither a stem. Okay, they don't have true roots, true stems, and true leaves, whatever it is, right? So they don't have all these things. Telephytes don't have all this, right? Okay, now we will actually discuss about one of the very important characteristics that I want you to focus, and this is very important uh, 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 for exam. Okay, please do concentrate over here. Now, if you see, we will talk some of the important characteristics. Okay, more about moles. Okay, okay. Yeah, talking about moles, these actually can be divided into two groups. Moles can actually be divided into two groups. What are those? They are septate and non-septate or coinocytic, that is same. Okay, now moles can actually be separated into two groups, or say divided into two groups. One is septate and the other is non-septate. You may know that septate basically means, if I just draw over here septate. Okay, this is a cell and this is nucleus. Okay, this dot one is nucleus and this is actually a cell. So you can see there is a cross wall. Okay, what is this? This is a cross wall. Okay, so this is actually septate, means like this. Okay, septate means what? They actually have cross walls. But if we talk about non-septate, it is like this. There is no cross walls. Okay, now the interesting thing that I want you to know over here is, see, if it is like this, there is no cross wall, so it is actually multicellular. Okay, means there are many nucleus over here, right? They are scattered throughout, right? This is called multicellular, and since there is a cross wall over here, it is called unicellular. Okay, simple as that. Okay, this is multicellular and this is unicellular because in each cell there is one nucleus. Okay, simple as that. Now, here comes the tricky part. That is about where is that? Here comes the tricky part, and that is about. Oh, just give me a second. Okay, let me just correct this one color. Okay, okay, we'll not work on it. Okay, you can see here reproduction. Okay, we will talk about the reproduction part. Now, how do these moles, or say the reproductive characteristics of these accepted and non-accepted, I mean, what actually do these accepted and non-accepted play roles? Okay, now it's a little bit tricky part, please do concentrate. Okay, please do concentrate over here. Yes, I'll just do like this. Yeah, so we actually, if we talk about reproduction, we can actually classify it or say divide into two types. One is asexual and the other is sexual. Okay, asexual and sexual pores. Okay, yeah, and sexual pores. Now, these asexual spores are actually called as what? Imperfect, or say it as imperfecti, and these sexual pores are known as perfect. Okay, clear up to this much? Okay, now these sexual pores actually are means asexual spores actually are septate and sexual are non-septate. Are you getting what I'm saying? These asexual spores are septate and sexual pores are non-septate. Means if we try to represent them in a diagram, septate means it looks like this. Okay, we have cross walls, and non-septate means it looks like this. Scattered, the nucleus are scattered. Okay, please excuse me when I'm drawing. I'm not a good artist. Okay, right. So these are the things. Okay, reproduction. If we talk about the reproduction, the asexual spores are imperfect C, and they are septate. And talking about sexual spores, they are perfect and they are non-septate. Okay, simple as that. Now these sexual spores can actually be like if we talk about the organisms, okay, or say if we talk about yeah, so what are those two organisms that are like do sexual reproduction? They are oomycetes, okay, and the other is no, please for yeah, sexual spores means oomycetes and zygomycetes. Okay, this is actually the examples of whoever goes like sexual reproduction or sexual who actually have sexual spores. They are oomycetes and zygomycetes. Talking about asexual spores, we have what? Ascomycetes and basidomycetes. Okay, basidomycetes. Okay, so these if you like please forgive me for making it rough because I want to explain it in the simplest over here. Okay, so the sexual spores are oomycetes and zygomycetes. They have these sexual spores, but these ascomycetes and basidomycetes, they have asexual spores. What do I mean by that? We can actually reverse the process. I mean an ascending process. Like this oomycetes, what color should I use? Okay, I think I can use this one. So this oomycetes and zygomycetes, they actually have what? Non-septate. They are non-septate. Okay, and this sexual asexual spores, ascomycetes and basidomycetes, they are septate. Simple as that. Okay. Now this is like a very tricky part, and I hope that everyone have understood from my explanations. And if you are not understanding, please rewind back the video and please see it again. Okay, so we are going to the next part, and that is types of what? Types of asexual spores. Okay, we will be discussing about types of asexual spores. Now, what are those types? Okay, I'll just list it down. What are the types of asexual spores? We have conidia. Okay, we also have oidea. Okay, and last but not the least, we have sporangiospore. Okay, so these are asexual spores. Okay, one more, I think I left this one. This is chlamydospore. Chlamydospore. Okay, so these are actually the leaves of asexual spores. Okay, now if we see a little bit detail about this conidia, they actually are cut off. Okay, they actually are cut off from where? Conidiophores. Okay, this conidia are this is asexual spores, right? So these conidia are actually cut off from the sporangiospores. Okay, second thing that you have to know about conidia is it is not enclosed in any container type. Okay, that means it is open. There is no enclosing structures of conidia. It is kept open. Simple as that. But contrasting to that, this talking about these sporangiospores, they are actually enclosed. These sporangiospore are actually enclosed under sporangium. Okay, these sporangiosspore are actually enclosed under sporangium. So what is this sporangium? Sporangium is basically sac-like structures. Okay, so what is this sporangium? It is basically if I just write it down over here, it is sac like structures. Okay, so what is sporangium? It is basically sac like structures, simple as that. Last but not the least, this is a very important thing and interesting thing as well that I want you to know. Okay, this chlamydospore. Okay, just let's change color. Okay, yeah, so chlamydospore, what is this? Now when any reserve food, okay, any reserve food is stored in mycelium. Here comes part of reserve food. Okay, when any reserve food is stored in mycelium, what actually happened is that these or those cells started to swell up. Okay, swell. Okay, and it actually enlarged. Okay, now due to this presence of this reserve food, the mycelium started swelling. Okay, the mycelium started swelling, and because of which there is an enlargement in cell wall. Okay, let it be cell wall. Okay, and because of which it becomes resistance to what? Since it have thicker cell wall right now, it becomes resistance to unfavorable condition. Okay, okay, just wait. Okay, I'll just make it smaller. Okay, it becomes resistance to what? Unfavorable conditions, or you can say unfavorable yeah, you can say conditions or climate or whatever it is, right? You know what I mean, right? So this climate is the spore. These actually are formed when any reserve food is being stored in mycelium, and because of which there is a swelling in the mycelium, and because of which it becomes resistant to unfavorable condition. Simple as that. Okay, last but not the least, we have what? Oh, sorry, we can use this one with this color. Okay, yeah, we also have arthrospores. Okay, yes, we have these arthrospores. Okay, so what is this arthrospores? Actually, this arthrospore, okay, this is a spore. This is formed when hypha undergo fragmentation. Sorry, we can use this one. uh yeah, yes. Okay, so when any hypha, or say, yeah, when arthrospores, how it is formed? When hypha undergo fragmentation. Okay, when hypha undergo fragmentation, these arthrospores are produced. Okay, what is this arthrospores? It is basically, in simple words, we can say it is cells of hypha. Okay, simple as that, right? Now when this hypha undergo fragmentation, arthrospore are produced. And what is basically arthrospore? It is basically cells of hypha. Simple as that. Now since we have studied already the asexual spores, now it is time for us to study about, or say learn something about, yes, you're right, we'll be studying about sexual spores. Okay, yes, we will be studying now about sexual spores. Now, there can actually be like four types of, yeah, four types of sexual spores. What are those? We have oospores. Okay. We also have zygospores. We have ascospores. Ascospores. We also have what? Basidiospores. Basidiospores. Okay, so these are a list of sexual spores. Now, as the name says, everyone knows, right? It is from Oomachites. What color should I use? I can use this color, yeah. Okay, so oospores are from Oomycetes. Okay, whenever I like say oh much this oh I mean I just want to use that word. Okay, whatever it is. Right, so oospores are actually from oomachites, and zygospores are from zygomycetes. Okay, and ascospores are from ascomycetes. Yeah, what is that? Yeah, ascomycetes. Okay, and basidiospores are from basidiomycetes. Okay, so these are the spores that are being derived from this one, right? From I have already mentioned here. Now, if we talk about some of the general concepts or general things that you have to know about, this is like, you know, these oomachites, or say yeah, oomachites and zygomachites. Okay, they actually are aquatic, means they live in water bodies. Okay, now these oomachites and zygomachites, they are aquatic. Not only that, it is formed by the union of male gamete and female gamete. Okay, these oomachites and zygomachites are formed by the union of what? No, no, basically not zygomachites. Uh, these oomachites. Okay, just let me this one. Sorry for that. Okay, now these oomachites. Okay, you can use different colors for this. What color should I use? Okay, I can use this color. Okay, so this oomachites. Okay, it is formed by the union of male gamete and female gamete. But what is the tricky part over here is, let me tell you. Okay, these these male gamete, okay, these are actually small, and female gamete are actually large. Okay, okay, understood what I'm saying? These male gametes are small, but female gametes are large. Okay, there is oomachites, or say, yeah, spores are actually formed by the union of small male gamete and female male gamete. Female. Okay, I get you what I'm saying. Okay, it is formed by the union of small male gamete and large female gametes. Okay, I hope you understand what I mean. Yeah, so one thing more that you have to know about these zygomycetes is, I think I can rub this on. Okay, so one thing that you have to know about this zygomycetes is that they have tough wall. Okay, they have tough wall, which actually enables them to survive even under drying condition. Okay, now these zygomachites, they actually have tough wall. Not zygomachites, I must say, these zygospores. Okay, these zygospores have tough wall, and because of which it actually helps to survive even under drying conditions, extreme drying conditions, obviously, right? And talking about these ascospores, as it is produced by ascomycetes, they actually are septate. Now you may tell me, sir, you have already told me that the sexual spores are uh, non-septate. But how come these ascospores are septate? See, these ascospores, okay, like I have already told you that ascomycetes and bacillomycetes, they are asexual, okay, and they are yeah, these ascospores, they actually are involved in this one now. They are actually are asexual, right? So they are septate, and there is a main thing that I have already told you, right? Yeah, so these septate, these ascomycetes are actually septate, okay, and it is formed by the union of two cells. Yes, it is common. Okay, it is formed by the union of two cells. Okay, and what is basidiomycetes, or say basidiospores? What should we know about this one? It is very common. You know what is basidiomycetes? I mean, which group of organisms I'm talking about? It is like mushroom, okay, rust, and smart. Plant rust, plant smart. Okay, actually not human one. Everyone knows that. just jokes apart. Okay, so these bacterial spores are like being produced by mushroom, rust, smart, actually plants, rust, plant smart. Okay, so yeah, this is the thing. So going to next, we have still to learn something about molds, and that is I can use this one. Which one should I use? Okay, let's use this one. Yeah, so moles. There's something still that you have to learn about moles. These moles actually require less moisture as compared to bacteria and yeast. Moles actually require less water or say less moisture as compared to bacteria and yeast. Okay, and not only that, these moles are mesophilic. Okay, what do I mean by mesophilic? Means they live in they can live in a temperatures around 25 degrees Celsius to 30 degrees Celsius. Okay, so this is the optimum temperatures for their survival. Not only that, moles are aerobic. Okay, means they depend on oxygen for their survival. Okay, and they will actually favor acidic environment. Okay, okay, so these moles favor this acidic environment. This is going to be the most important part till now. Okay, this is very interesting, and at the same time I don't know whether you're interested or say on this food microbiology part, but believe me, I really like these chapters. Okay, now if you see the inhibitors, where should I write? Okay, I can write over here. We will be talking about the inhibitors. Okay, we will be talking about the inhibitors. Inhibitors of what? Moles. Okay, means these are actually the chemicals. You can no, not the chemicals. First of all, we will learn about this one, organisms. Okay, inhibitors that is being produced by a certain or say different types of moles. First thing first, you may have all heard about penicillin. Okay, you may all know this one, but penicillin is being produced by which moles? Okay, it is produced by Penicillium chrysogenum. Okay, this Penicillium chrysogenum produce the inhibitors, that is penicillin. Okay, and clavicin is another, clavicin is one more inhibitors that is being produced by Aspergillus clavatus. Okay, this Aspergillus clavatus produced an inhibitor called clavicin. Now we will learn about the chemicals. Okay, chemicals that is inhibitory to mole growth. Chemicals, okay, just this is not right. Okay, yeah, more we can write like this. Okay, mole. Okay, just excuse me. Mole growth. Okay, inhibitors. In fact, this is actually the chemicals. Okay, the chemicals that is inhibitory to mole growth. Now what are those chemicals? Okay, there is inhibitor to mole growth. They are like sorbic acid. Okay, sorbic acid and propionates. Okay, and acetates. So these actually are inhibitors. Okay, that is inhibitory to mole growth. Okay, I think that is enough for these inhibitors. Now still continuing the continue learning more about these moles, we can say this, that what is moles actually. If we be very I want to be very clear. Okay, that's why I just want to clarify this all these things. Now moles actually are a type of plants, to be very simple as that. I mean, yeah, it is actually a type of plants, and the kingdom of these moles is Mycete. Okay, the kingdom of moles is this kingdom. Okay, yeah. So the kingdom of moles is what? Mycete. And these moles, they don't have roots, they neither have leaves. So these two things actually are not present in moles. Not only that, they also don't have chlorophyll. Okay, they can't use sunlight for its food. Okay, and they belongs to what? Eumycetes. These moles belongs to Eumycetes. What is Eumycetes? It is a true fungi. Okay, okay. So they belongs to these moles actually belongs to Eumycetes. Okay. Now one more thing that I am not still not talking about is about very common thing. If you are from agriculture or yeah, from agriculture background, you may know this one. Or yeah, so this is what? Rhizopas. Okay, what is this rhizopas actually commonly called as? It is called as what? Bread mold. Okay, we can see this one in bread. It's very common actually. Okay, so this is actually a bread mold. Now this is going to be again very important, like a pigmented type. Okay, now we will actually see different colors of this one. Okay, now we will see this one. Just give me a second. Okay, we can go to next page. Okay, yeah, so we will learn about blue contact moles. We will also learn about pink moles. We will also learn about dairy moles. Last but not the least, we also have red bread moles. Okay, so these are the different types of moles which are actually of different colors. Okay, yeah, so these blue contact moles, which mold is that? It is actually Penicillium italicum. It is called as blue contact moles. And what about pink mold? It is Trichothecium roseum. Okay, it is Trichothecium roseum. And what about dairy molds? It is Geotrichum candidum. Candidum. Okay, this dairy mold is actually what? Geotrichum candidum. And red bread mold is Neurospora cytophila. Okay, yes. So this red bread mold is actually Neurospora cytophila. Okay, yeah, so what do we have next? Okay, so one more very important thing or very interesting thing at the same time, I must say, every time I say this one because it's my favorite chapter after all. Okay, now about this sporotrichum carnis. Okay, this sporotrichum carnis, what is this and what it causes? Okay, this sporotrichum carnis basically it causes white spot. Sporotrichum carnis cause white spot, but where? In chill meat. Okay, this Sporotrichum carnis cause white spot in, where should I write? Here. This is not good. Okay, I can use this one. Yeah, they cause white spot in chill meat. Chill meat means fresh meat. Okay, so this Sporotrichum carnis cause white spot in chill meat. Simple. Now we are over with the molds and we are now coming to what? We are coming now to, yes, we are coming now to yeast. We'll be studying about yeast now. How do I make it more beautiful? Okay, okay, so we'll be discussing about yeast over here. uh Yeast, we all know that yeast is actually not filamentous. Okay, okay, they don't have filaments. Okay, whatever it is, and they are unicellular. What do unicellular looks like? hmm? Unicellular looks like this. This is the nucleus, and yeah, so this is how unicellular looks like. Okay, yeast are unicellular. Okay, and they reproduce by fission. I have already told you, they reproduce a fusion and budding. Okay, so yeast actually reproduce by fusion and budding, simple as that. And the fermentation, when they involved, when are they involved in, whenever this yeast involves in fermentation, they will give a this one, bread, wine, beer, and vinegar actually is the product. Okay, simple as that. This is very simple concepts that you already know. Okay, now one very important thing, interesting thing about yeast is actually there is one more called as false yeast. So what is this uh, you know, false yeast? These false yeast, these actually are not produced by ascospores, don't produce ascospores. Okay, so these false yeast actually are don't not produced by, okay, just wait a minute. Okay, ascospores, ascospores. Okay, we will learn about this one in the next chapters. I mean, not next chapter, in the next uh, page. Okay. Okay, so talking about more about yeast. Yeast actually are what they are. They can be oxidative and fermentative. Okay, so this yeast can actually be oxidative and fermentative. Now what are those, what do they means, we will actually learn right now. Okay, now these oxidative, okay, what happens in this oxidative is like uh, they will form film like, okay, this is the film like on the surface. Okay, they will form like film, or say where should I write this one? Okay, I can write over here. Okay, they will actually form a film like I think this is, okay, so these oxidative yeast, they actually form film like, or say scum like, or say pellicle. uh what is this actually? Please excuse me with my handwriting. Okay, now what actually it means. Basically what happens is that when these oxidation undergoes, okay, because of the production of means whenever this undergoes oxidation, uh, this yeast actually comes up on the surface. Okay, and it forms like a film. Okay, and because of which it gets its name from the process, right? It is called as a film forming this one, or pellicle, or scum, whatever it is, right? So this in oxidative, it comes in the surface. That is the only thing that you have to understand and know. But this fermentative, they grow throughout the medium. Okay, let it be like this. Okay, so this fermentative actually grow throughout the medium. And in fermentation, there's also a production of carbon dioxide. Okay, oxidative yeast, they will grow only in the surface, but and of course or say form film like structures. Okay, film like covering, whatever it is. But in fermentative, they will actually be growing throughout the liquid. So, and there will also be a production of carbon dioxide. Okay, now yeast actually require uh okay, I can write here. Yeast actually require more water as compared to moles. Water in terms of, like, in say moisture. Okay, 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 so yeast actually require more water or more moisture as compared to moles. Now yeast is also a mesophilic, means they can live in the temperatures of this is a favorable temperature. Okay, 25 to 30 degrees Celsius. Okay, this is And not only that, they are also aerobic, means they require oxygen, air for survival. Okay. And the yeast, where do they get their energy from? This is also very nice thing that you have to know, right? So they get energy from sugar. Okay. Yeast actually get energy from sugar. Okay. Now we will learn about classification of yeast. Okay, please bear with me. My spelling, my handwriting is not that nice, I know. Okay. uh, we learn about yeast classification. Okay. Yeah. So yeast classification, it's very easy terms. Okay. In this yeast classification, what are they? We have true yeast, and we have what, false yeast. Just contrasting one. Okay, we have true yeast and false yeast. Now what is true yeast and what is false yeast, sir? Okay, don't worry, I'm here to explain you, right? Now this true yeast, they are from subdivision what, Ascomycina. Okay, they are from these subdivisions. And false yeast are actually from subdivision Deuteromycotina. Okay. Now true yeast and false yeast, are you understand what I'm saying? True yeast are from actually from the subdivision Ascomycina, and false yeast are from the subdivision Deuteromycotina. Hmm? Okay. Now talking about false yeast, they are actually most of the time sporogenous. Okay. Now these false yeast are from like, I mean, they are sporogenous. What do I mean by that? It's basically uh wild yeast. Okay. Yeah, wild yeast. Now you tell me, sir, what is wild yeast? What do I mean by wild? Wild yeast actually means, like, they are not being used, or it's not our concern, or it is not being used for any kind of human welfare. We don't actually use this one. Suppose we are using one yeast and we don't use another. Now the another which we have not used, that becomes a wild yeast. And it is called asparaginous. Okay, that is the only thing that you have to know. That's a simple concept. Okay. Now, talking about asexual reproduction of yeast, what are the different methods, or say, yeah, how do asexual reproduction takes place in yeast? Asexual reproduction in yeast. Okay, asexual reproduction in yeast. I think I can write it a little bit better. Okay, yeah. So asexual reproduction in yeast, what are those? We will just list it down. We have budding. We also have fission. Okay. We also have okay, so these are the different types of asexual reproduction that yeast undergoes. Okay, budding and fission I have already mentioned. I didn't think why I'm mentioning it again. Okay, so whatever it is, these are the types of this one. Okay, asexual reproduction of yeast. uh, one more concepts that you have to know about yeast is about the this one. What do I have? I have this one, top yeast and bottom yeast. Now what is this top yeast and bottom yeast? Okay. Now if you see top yeast means it is at the top. What happens? What happens? I will tell you. Now in top yeast, what happens is that there is a clumping of cells takes place. Clumping means together. Okay. Now binding of these cells take place, and the production of carbon dioxide also takes place, because of which it started floating above. What? Because of which it started floating above, and because of which it is called as top yeast. Are you getting me? The first of all, the cells get clumped up. Okay, the cells get clump means getting together, right? And the production of carbon dioxide also takes place, because of which all the cells come up in the surface, or say on the surface, and because of which it is called top yeast. And what is bottom yeast? Bottom yeast, in bottom yeast there is no clumping of cells. The cells are not coming together, and there is no production of carbon dioxide, because of which the cells got a chance to come and settle down, and because of which, or say settle bottom, in the bottom, and because of which it is called bottom yeast. Simple as that. I hope you understand the concepts that I mean. Okay, now very important thing, please concentrate here. Okay, hello, are you listening to what I'm saying? I am talking to you. Please do respond. Are you understanding what I'm explaining to you? You are still not speaking, yaar. कुछ तो कहो यार. Are you understanding? Okay, come on. Jokes apart. Let's see this one. Okay. Yeah. So yeah, these top yeast actually are. You know what? They actually are what? Active fermenters. Okay. These top yeast actually are active fermenters. And they actually grow rapidly at how much? 20 degrees Celsius. Okay, these top yeast actually are active fermenters, and they grow best at, or say rapidly at, 20 degrees Celsius. Simple as that. I'll change the color. Okay, so it becomes a little bit more tricky to the eye. What can I use? Can use this one. Okay, and about bottom yeast, these bottom yeast actually are the what? It is the best fermenters. Best fermenters. Okay, understand? Top yeast actually are active fermenters. They grow rapidly at 20 degrees Celsius, and these bottom yeast are the best fermenters. Okay, okay, simple as that. Now, last but not the least, we will learn about some of the techniques. What is the techniques, college? We will learn about uh, pure culture techniques. Okay, we will learn about what? Pure culture techniques. Now, what do I actually means by I can change the color. Should I change? I'll keep it like that. Okay. Yeah. So what do I mean by pure culture technique? Pure culture means what? There is only one species or one type of organisms. Okay. Okay. This is actually pure culture. It means pure, right? There is no contaminations of, or so there is no mixing of different species of organisms. Only one type of organisms or one species of organism. Okay. Now, different types of pure culture techniques are like this. Not one. Okay. This is the thing. Aseptic techniques. Okay. These are all what I will be listing down right now is actually all these pure culture techniques. Okay. Next we have stick methods. We have stick methods, and third we have pour plate methods. Fourth we have what? Spread plate method. Okay, spread plate method. Last but not the least, what we have is, yeah, we have serial dilution method. Okay, yeah, so these are different techniques of pure culture. Okay, I mean, to obtain a pure culture, these are different techniques. I just want to add something here, that is about ascetic technique. Okay, so what is the main objectives of aseptic technique? To be very specific, there are actually two objectives. Okay, one objective is to obtain pure culture. Just let me make it. Okay, so the one objective is to obtain pure culture, and second is to prevent any contamination. Okay, so these are actually the two objectives of aseptic technique. One is to obtain pure culture, okay, and the other is to prevent cross contamination, or so to prevent contamination. Simple as that. Now we will learn each of the techniques I mean, not that in detail, but we will try to understand as much as you have to understand, okay? Not less and certainly not more. Okay, so we'll start with the first, and that is what color should I use? I can use this one. Okay, we'll start with this one. Okay, we'll start with what? Aseptic technique. TCHNIQUE. Okay, yeah, so this we will study now about aseptic technique. Now this aseptic technique, as I have already told you, that there is two objectives, right? One is to obtain pure culture and the other is to prevent cross contamination, right? So these objectives actually are obtained or maintained by using different methods. Okay, by using what? Different methods. What are those different methods? Yeah, so they are like flame sterilization, uh, tube transfer, tube transfer, okay, and what we have spread plates also, spread plate, okay, we have pour plate, right? So these are actually all the different methods. Or one more is that about stick plates, okay. So these are actually the different methods that actually are used for this aseptic technique to obtain those two objectives. One is obtain pure culture and the other is to prevent cross contamination. Okay, now we will learn about stick method. Okay, where is this? Yeah, we will be learning about stick methods. Okay, now talking about this stick methods, this stick method is done to what? This stick method is done to yeah, I can use this one. Is done to obtain the isolated colonies. Okay, this stick method is used to obtain these isolated colonies. Not only that, this stick method is very rapid and it is simple at the same time. Okay, sting methods are done to obtain the isolated colonies, and it is very simple and rapid. Okay, what actually we do in sting method is dilution. Dilution of what? Dilution of sample or inoculum. Okay, now what do I mean by dilution? I hope everyone understand. Dilution means basically reducing the number of microorganisms, okay, by serial dilutions, okay, by yeah, in simple terms you can understand, like we are reducing the microorganisms, okay, as simple as that. uh I will explain you all this in serial dilution method, okay. Come on, now let's go to pour plate method. Okay, one more method do we have, and that is this one. What? Pour plate method. Yeah, so pour plate method. Now it is also here also we dilute the samples or dilute inoculum, okay. Excuse me. Yeah, so here also we dilute the inoculum, okay, and in tubes actually we dilute, that also we do in tubes, but the important thing over here is that we dilute in the medium, and that medium is actually liquid, or you can say melted what should I write? Liquid agar medium. Okay, now we dilute, okay, we do dilution of those microorganisms in a media, and that media is actually liquid agar medium. Liquid means it is being melted. What happened actually is that, about this agar medium, one interesting thing about is that it is this liquid, it becomes liquid only at 42 to 45 degrees Celsius. Okay, this it becomes, this agar medium is liquid on this temperature. Otherwise, if it is below 42 degrees Celsius, now it will become solid. Okay, this agar medium will become solid if it is at 42 degrees Celsius. I mean, or I mean below 42 degrees Celsius. Okay, in the range of 42 to 45, it will be yeah, this one liquid. Okay, that time we can do this pour plate method. Simple as that. Now, why do we use this pour plate method? Is basically to determine what? To determine number of viable bacterial cells. Okay, so you have to understand this one. This is very important. Okay, just let me mark this one. Yeah, I can use this one. Okay, why is a pour plate method used? It is used to determine the number of viable bacterial cells. Okay, so this is a main objective, you can say. And in this pour plate method, what I mean is that only the I can use this one. Yeah, only surface colonies develop. Only surface colonies develop. Okay, okay. So in pour plate method, only the surface colonies do develop. Simple as that, right? Can I take it a little bit? Okay, okay. So what happened is that only surface colonies developed in this pour plate method. Last but not the least, we have one more, and that is what? We have spread plate method. Okay, okay. We have spread plate method. Okay. What do we have to know about this method? Yeah, I can use this one. Okay. What do we have to know about this one is, um, here also we do dilution. What? Here also we do dilution. But the contrasting thing about this, or sorry, different thing about this dilution is we do in a medium that is sterile. Okay, we use sterile medium. But if you see here in the pour plate method, what do we use? We use liquid agar medium, right? But here, uh, what do we use is actually sterile medium. Sterile medium means what? Like you can say you can use water, or maybe we can use a saline. Okay, yeah, so these are the medium that we can use in dilution. Okay, simple as that. Now the thing, yeah, so the last but not the least, one more this one we have, what is that? Methods, and that is serial. I hope everyone know this one. Serial dilution methods. Okay, we have this serial dilution method. Yeah, so why do we use this method, and what is its what is that, usefulness? Okay, utility, we will see right now. Okay, so serial dilution method, actually this type of dilution method is used for those organisms. Okay, this serial dilution method is actually used for those organisms that are not yet cultivated on solid media. Okay, they are actually growing on liquid medium. Okay, but till now they are not actually grown on a solid media. Okay, so this is where we use a serial dilution method. Nothing much to know about this, you know I just have to be, your basics have to be clear, that's all. Now last but not the least, one more thing that I want to say about this one. Where is that? I want to change the color. What color should I use? I can use this one. Okay, now one more thing that I want to say about is that about this one is about maintenance. Okay, maintenance and preservation. I don't know whether you know this one. but this is very important. Okay, like maintenance and preservation of pure culture. Okay, in order to maintain those pure culture that you have made, or say the you that you have isolated. Okay, it is very not easy to preserve and maintain those pure culture. Why? Because these culture, they used to grow. Okay, they grow, and there is a need of subculturing. What do I mean by subculturing? It means basically you have to change it to a new media. Uh, 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 Continuous checking and continuous maintenance, continuous transferring to another media is actually required. Okay, and it is time consuming, and it is very not it's cumbersome job actually, right? So maintenance and preservation of this pure culture is not that easy. Now, in order to tackle that problems, we have developed some of the uh, modern methods. Okay, some of the modern methods. What are those? Let's see. Those modern methods. One of the modern methods are refrigeration. Okay, and another one is paraffin method. Okay, paraffin method. Okay, and cryopreservation is also one of them. hmm? Modern methods of maintenance and preservation of pure culture, right? Last but not the least, we have lyophilization. Okay, so these actually are lyophilization is known as freeze drying. I just forgot to mention this one. You have to know this one. Okay, okay, so these actually are different types of modern methods that actually helps to help in maintenance of the pure culture, or say preservation of the pure culture. Okay, now we will try to understand each one of them in details. If I can see finish here, let me see. Okay, now see refrigeration. Refrigeration is basically, we know it is in cold temperature in fridge, right? So what is the temperature that is being maintained over here is around 0 degrees Celsius to 4 degrees Celsius. Okay, so this is the temperature that is being maintained. Okay, 0 degrees Celsius to 4 degrees Celsius. Not only that, for bacteria, it is kept in these temperatures for 2 to 3 weeks. Okay, for bacteria, how much it is? 2 to 3 weeks. And why are you not responding? Please do response. Please do response. I know you are just looking at a video. Please do response. Okay, whenever I ask, please do answer, because I may not able to hear you. But if you do response, it will keep your concentration active, and not only that, you will also remember and understand at the same time, right? So please do response. Okay, don't think it is only a one way of communication. Try to make it from your side two way of communication. Okay, so that our sort of like lectures could be two-way communication, and you could understand better and retain the information that I have shared with you. Right? So please do response. Okay, if you don't respond, you are cheating yourself. Don't do that. That's not good. Okay, yeah. So talking about this fungi. Okay, so for this fungi, how much duration should we keep? Again, you are not just listening, you are not responding to me. Please do response. Okay, please do response. I know you are not responding to me. Now this fungi, they can uh, they have to be kept for 3 to 4 months. Okay, they have to be kept for 3 to 4 months. Okay, they can be kept for 3 to 4 months without any maintenance in this temperature. Okay, okay, yeah. So we will go with the next method, that is yeah, it is paraffin method. Okay, paraffin method. Okay, you can change this one right now. Okay, I'll use this one. Okay. Excuse me. Yeah, so this paraffin method is actually the simplest and economical at the same time. Okay, so this paraffin method seems to be very good method because it is simplest and economical. The returns, or say the investment that we have to use, is very less. In investment is very less in this paraffin method, so it is a very good method, right? Yeah.